Welcome to Pharma-info with Shubhashish. Today, the topic which I have brought for you, that is drug design, right? Actually, prompted by the PCI syllabus and all, because in UG PCI syllabus, basically in 5th semester, they have kept a module, okay, which involves the strategies used in drug design and pharmacophore modeling, the molecular docking study, then combinatorial chemistry. So these are the things they have kept, right? So this discussion is basically I'm going to segregate this particular discussion in so many parts so that and you can follow them sequentially, right? Because completing a module of 7 hours is not possible in a single video, right? So that is why, so whatever thing that I'm going to deliver right now, so that I'll just split in several parts and all, and each part I'll upload for you so that you can go through it, right? Now, so and moreover, let me just tell you, because as they are asking, as this is at the level of UG, right? So that means I should not discuss each and everything of drug design, right? So whatever thing that is there in your syllabus, and basically I'll just stick to the time also, right? So that without dealing without making it more extensive, okay, let me just highlight the areas which need to be focused, right? The areas which need to be touched, right? So now drug design, now the objective of this particular discussion is here I'm going to discuss the strategies, then your strategies of drug design, right? And then pharmacophore modeling, then your molecular docking, Now, before pharmacophore modeling, I'll discuss QS here as well. And once we are done with this, all these 4 different things and all, then we'll move to combinatorial chemistry, right? Combi-chem, right? So now let us just begin the topic with the strategies, okay, which are usually being adapted in drug design, right? Okay. Now, we all know that drug design is a very broad topic, right? It's a very broad topic, right? Drug design. And what for we are designing a particular drug? See here, intentions are found to be different. Sometimes we are after, developing a lead. Sometimes we are after finding a lead, right? And in search of a lead, drug design may be opted, right? Sometimes we have with us the lead. Only thing is that we need to just make the lead more optimum, right, for the therapeutic effect and all. Sometimes it may also happen that we don't have any receptor, so just to explore the receptor, right, we are opting this drug design and all, right? So there are different areas or different areas where drug design is found to be opted or adapted, right? Okay, or we can say that so let us just deal with them one by one, the different strategies which are being adapted, right? So if we just try to generalize it, if we just try to figure it out, that the strategies includes what, or what are the different strategies which are adapted in drug design, right? We have with us basically, one is your SBDD, or we can say instead of S we can write M, right? M. So if we just expand this acronym, right? We just try to expand this acronym. It stands like macromolecule based drug design. So it's M for macromolecule based drug design, right? Okay. Macromolecule based drugs. So what are macromolecules? So macromolecule is nothing but the receptor or the enzyme, sometimes, or the DNA as well. That means the genetic material, right? DNA or RNA, anything, right? Now, shall we take them in raw form and just put inside the computers? It's not like that, right? So that means we don't have to deal with the real receptor or the real enzyme, but the real enzyme which has been elucidated out of X-ray crystallography or NMR. That thing only we are going to study, right? So that means here the receptor, enzyme, DNA, we are going to deal with their crystallographic form, right? We are going to deal with their three-dimensional form, and that too a crystal, right? That means we have to deal with the atomic resolution structure of the receptor, enzyme or DNA, right? So here macromolecule based, what does it mean? Or structure based. Okay, why I have written macromolecule? Initially I have written structure, right? But structure can be a macromolecule, it can be a small molecule as well, right? So we can say instead of putting S, we can just write MBDD, macromolecule based drug design, right? Because structure based drug design, if we just try to figure out structure based drug design, this can be split into two major types. One is your macromolecule based, the other one is your small molecule, often called ligand based, right? Ligand based, right? Okay, so chalo, let us see one by one. So now we are dealing with what the strategies, right? So one of the strategies found to be macromolecule based drug design. The intention behind drug design is found to be clear: in search of a lead, in order to optimize the lead to a certain extent, in order to explore the active site of a receptor. This can also be used, right? Okay, chalo. But the basic thing is that in search of a lead, in search of a novel compound, in search of a novel compound, drug design strategies are being adopted frequently, right? And that is nowadays, that is not a traditional one. Nowadays, as with the advent of computer and the softwares and the programs, right? Now system is found to be more improved or upgraded. So that is why we are making it in silico. So what are the things that we are going to do? That is in silico only. That means through computer. That means computer based or computer aided or computer assisted drug design, right? Okay. Under that, all the things will come, right? We'll see later on. Okay. We'll see sequentially rather. Now, so macromolecule based means first and foremost we need to collect what? We need to collect the receptor or enzyme or DNA, okay, of query, right? So this is the thing that you have to collect from where? From what? From a data bank, like your blood bank. Okay, these people are just storing or preserving the blood, right? Where? In the blood bank, right? And in each and every bottle they're just putting what? The group, right? So that they can readily identify that one and give it to the person who is after that, right? Who is seeking that one, right? Or who is in need of that particular group of blood, right? Chalo. Now same thing here also, as they are proteinous in nature, except DNA, receptor, enzyme, both are proteinous in nature. That means the atomic dissolution structure comprised of what? Multiple number of amino acids. So all amino acids are joined together so that there is a formation of a protein, right? Okay. Now before that, I strongly recommend please go through each and every amino acid that is there in our biochemistry, right? So only 20 are there. Please go through it. Get just familiar with the structure and all, familiar with their properties and all. This may help you in dealing with the drug design in a convenient manner, right? Okay, sure. Now the thing is that, so one is receptor, the other one is enzyme, and the final one is DNA based, right? DNA based drug design, right? So all are found to be an example of macromolecule, right? Macromolecule. So macromolecule based drug design, how this can be done? So once you are collecting the receptor or the target from the protein data bank or the data bank, okay, then you have to study that particular macromolecule, right? Study the macromolecule in a suitable platform, right? In a suitable platform, there is n number of softwares are there through which you can visualize the protein, right? Visualize the protein collected from the protein data bank, right? Chalo. So now the thing is that, so here macromolecule based means once you are going through the structure, three-dimensional structure of the macromolecule, the next thing is you have to find out the active site, right? How the active site can be found out? The active site can be found out once you are taking the protein, considering the fact that the protein is already bound to a particular ligand, or rather a ligand bound protein. That means a co-crystallized structure. It is more preferable that if you, instead of taking a protein, unbound protein, you just go for a bound protein, right? You can get n number of protein there in the protein data bank. Say we have few sites are there, Out of that, two are found to be very important. One is your RCSB and the other one is your NCBI, right? So these are the two sites. Just in Google type RCSB, it will take you to that paper, to that particular page where you can just search your item. Search your item means suppose ki you are in the process of developing anticholinesterase, right? So that means here the cholinesterase enzyme is found to be the target for you, right? Just putting cholinesterase in that particular port, right? In that query port, okay, it will just deliver you so many proteins, right? And out of the entire set of proteins, right, you have to search for the protein in which the active site is bound, is already occupied with the ligand. Got my point? So then only you can readily readily trace, yes, this is found to be the active site. Because until and unless you are not you are not acquiring the idea about the active site and all, it's very hard to proceed further, right? So that means in order to proceed further, it's better or it's been wise of you if you just go for a protein which is found to be occupied with a ligand. That means ligand bound protein should be collected from the protein data bank. From where you can get those things? RCSB and NCBI, right? Sure. So now once you are done with this active site recognition and all, okay, then what next? The next is you have to, as I said, that the one that you have collected is found to be a ligand bound, right? So that means both are in their 3D fashion, right? And definitely we are expecting that the ligand, while being occupied within the active site, definitely it will it definitely form the suitable interaction with the active site residues. We are not expecting that all the amino acids within the active site of that particular receptor or enzyme is involved in making an interaction. It's not possible, right? Because the ligand which we are dealing with is a small molecule. It's a small molecule, right? So that means few functional groups are there. That means with those functional groups only we have to find some kind of interactions and all, right? So that means suppose ki one ligand is having 4 functional group. That means 4 max 4 interactions are possible, right? So that means 4 amino acids within the whole active site, they are involved in making an interaction with the ligand. Followed, na? Chalo. Now the thing is that so that means after tracing or tracking the active site, okay, the next thing is that just focus on the ligand, right? Focus on the ligand and the way it interacted with the active site amino acids, right? So that means two things you are done with 2 things. One is you have identified the active site. You have identified while going through the binding mode, you have identified what? You have identified the active site residues which are participating in the process of interaction, right? And moreover, the active functional groups which are present within the ligand. So that means you have collected the feature you have with you the information of the active site, at the same time the information or the features or the key features, right, which are responsible for the interaction of that particular ligand, right? So that means you are done with what? You are done with the ligand, at the same time the active site, okay? Now what next? That means as I said that drug design, the intention is to develop a lead, right? Either to develop a lead or you have to optimize the existing lead. Consider the ligand which you have interacted just now, which you have found just now within the active site, right, is a good ligand, is a good lead, right? Is a good lead. So that means definitely all you want is to optimize the lead further. So that means, and now you are done with the functional group. You have already traced the functional group present in the ligand. Now what next? The next is just do some kind of permutation combination within the lead, right? Make some new lead and just dock it inside the active site and just get the binding energy. You can get to know whether they will fit or not, right? But make sure that while making that particular drug, considering the topology or considering the feature of that particular ligand, that is the co-crystal, okay, just make sure that they should be synthetically feasible. Right? Okay. And for that you have to be thorough with the different organic reactions and all, right? Okay. Then only you can have an idea, okay, chalo, whatever structure that I am going to, build in pen and paper, okay, are they synthetically feasible or not, right? Okay. So that means docking, right? So that means in case of macromolecule based drug design, target identification, lead identification and lead optimization, and then get the interaction profile through molecular docking. So this is the sequence that has to be followed in macromolecule based drug design. Got my point? Chalo. So this is one of the strategy, that is macromolecule based drug design, right? Chalo. So with the help of this macromolecule based drug design, that means here we are bringing the method called molecular docking, right? Molecular docking means you just want to know whatever the ligand which you have just made in your paper, right? And after getting the three-dimensional structure of the ligand, whether the ligand is suitably placed within the active site or not. Okay, is it forming some kind of interaction with the active site amino acids or not, right? That you want to see through molecular docking. So this is the purpose behind molecular docking, right? Chalo. So this is your macromolecule based drug design, or it comes under structure based drug design. The next thing is your small molecule. That means ligand based drug design. Ligand based drug design. Chalo. So ligand based drug design means QACA. So here you have to deal with what? Here you have to deal with the small molecules. That means the ligand. That means the ligand, right? That means the molecule or drug molecule, we can say. In a straight way, drug molecule, right? So QACR, under QACR, basically we have to focus on 2D and 3D. But in your syllabus, as I said that this particular discussion is I'm just strictly following the UG syllabus and all, right? So that means I'm not supposed to discuss each and every facets of drug design, right? So in your syllabus, only 2D QACR they have given. They are asking to study this 2D QACR, not 3D QACR. And let me give you for your information only, we have QACR starting from 1D to 6D. 6D, okay? It's not that we are adding an extra dimension to it, right? Dimension with respect to a feature, right? Dimension with respect to a particular feature. Dimension means it's not that XYZ. Okay, up to 3-dimensional XYZ. We are adding Z so that the 2D has become 3D. X, XY, and then 3D means XYZ. After that, beyond that, we don't have any other choice, right? But we can add some extra feature which we are considering it as an extra dimension. We are just opening up an extra dimension of that particular process, right? Of that particular event, right? Chalo. So now the thing is that 2D QACR, 3D QACR. Now in your syllabus, in PG or according to PCI syllabus, right? They are asking for this 2D QACR and they are asking for few descriptors, right? They are asking for few descriptors, 3 to 4 descriptors they are asking, that we'll see. And after that they are also asking Hans analysis, right? Hans analysis. Or Hans equation, right? Hans equation. We'll see what exactly Hans equation means, right? So in QACR exactly what we are doing. In QACR exactly what we are doing. Say, as I said that 2D QACR means 2-dimensional QACR, right? QACR means what? Quantitative, structure, activity, relationship. Okay. Now, let us just see that what is ACR. ACR means, we all know ACR, right? I have 6 to 7 videos there in my channel, right? That is pertaining to ACR only, right? In fact, today also I have just published one video, ACR of barbiturates, right? If you have time, you can go through it, no problem at all, right? Now, so ACR means what? ACR is nothing but it's a qualitative check. That means here also we have to deal with so many structures, right? But what is common in them? They are sharing a common template. Suppose they are sharing a common body. Only thing is that we are just changing the hands, the legs, the heads, something like this, right? So that means we are just considering this modification process, and the weapons that we are bringing in this modification process, they are nothing but the substituents. So what we are exactly, the statement that we are putting here, insertion of a particular group at a particular point of that central body, whether it elevates the activity or it deteriorates the activity. Whether the activity abolishes or it improves, that we are just looking after through ACR. It's a qualitative check. Now, with this ACR, we can raise a question that why this functional group? Why this change? Why this change of functional group brings about a change in the biological response, right? Why? And what for you are getting the biological response? Why the biological response is elevated? That is simply with this substitution. Why it is elevated? Why it is elevated? Because definitely the contribution is coming from the additional contribution is coming from that substituent. Okay? We just replace the old one and just try to fix with the particular template, the existing one, right? So that means we are just changing the old one with the current one. right? And the current one should have the potential to elevate the activity. That means it is with the help of this functional group the activity elevates, right? It's not remains same. We are dealing with a particular event where we have found that insertion of a chloro group at 7th position of benzodiazepine leads to increase in activity, CNS depressant activity. What does it mean? That means earlier it was unsubstitution. They got an activity, right? They got an activity. But the moment they inserted one chloro or electronegative group at 7th position of benzodiazepine, which eventually leads to elevation of CNS depressant effect. That means maybe it is the chlorine that is contributing. As an electronegative atom, it should contribute electronically. It should contribute electronically. It may happen that it is the chloro, by virtue of its electronegativity, it is forming an extra interaction with the active site residues. Now, why we are calling it as active site residues, despite of the fact that this protein comprised of amino acid, but still we're just calling it as residues. Why? Because residues. Why? Because amino acid, while being a part of a protein, loses its partial structure. At least a portion of that particular amino acid will get used up. Right? So it is a portion because it has lost in the process of what I should say, in the process of making a protein. It is the amino acid. Each and every amino acid has to lose a certain portion of it so that they can connect to each other. Am I correct? Suppose we are using the C terminal of one amino acid, we are just connecting with the N terminal of another amino acid. Can we say after getting this CONH, can they bind in this form? No, it's not possible. That means on removal of water molecule only, there is a formation of a peptide linkage, right? So how this peptide linkage has been formed? It is out of release of a single molecule of water. That means a portion has been clipped off. A portion of the amino acid has been clipped off. That means it has lost its uniqueness. Now we cannot call it as, yes, glycine is here. Yes, we can say a portion of glycine is here. Can we say glycine is here means you can find glycine as a whole, but the whole thing is missing here. So that is why instead of calling it as amino acid, when it is a part of a protein, right, you should call it as a residue. That means the residual matter is left here. You are just observing the residual matter, not the whole thing. That is why we are usually calling it as the residues, the active site residues. So that is why we are calling it as residues. It's not that okay, chalo, residue means it's synonymous. It's not synonymous. Residue and amino acid both are not found to be same. Residue means a portion of amino acid. Amino acid means it's a whole amino acid. It's a zwitterand, right? Right? It's a zwitterand. Chalo. Now, so where exactly we are? We are in Asia, right? We are in the search of, so why this, why there is an increase in activity? Why there is a depletion in activity? That means the functional group which came in that replacement process is responsible for either elevation or deterioration, right? Right? So if that is the case, that means probably it is the property of that particular functional group that is responsible for the elevation, for the deviation, either positive deviation or a negative deviation, right? Chalo, thik? Now, here comes, so that means it is the biological activity that relates to the property of the functional group. The property of the functional group. Which property? Electronegativity. Which property? Chemical property, right? Sterically bulkier. Which property? That means we are dealing with the volume, shape and all, physical property. That means it is the physical as well as chemical properties. They are altering the biological response. So this is the thing. If we just try to correlate them, biological activity depends on what? Depends on what? The physical as well as chemical properties of what? Whether the whole drug? No. Of the substituent. Because it is because of that particular substituent only we got an elevation. So that means no longer we should say that it is the whole compound. Rather, the substituent's contribution. It is because of the substituent's contribution we have found this alteration. It's not that the whole compound you are changing. You are changing a portion of the compound. You are changing a portion of the compound, that too it is substituent, right? So that means it is the substituent that is contributing. So it is the contribution of the substituent that has to be taken into account, right? So we can say it is the biological activity that depends on what? That depends on the physical as well as chemical properties of what? The substituent placed in that particular molecular skeleton. So this is all about QAs here. This is all about QAs here. This is all about QAs. Here comes the concept of QAs here. There we are just quantifying the structure-activity relationship. How these properties, how these parameters, how these descriptors are affecting, are influencing the biological activity. Okay, that we are looking after. So it is about the small molecule or ligand-based, right? We have another thing apart from this QAs here. We'll see about this QAs here, right? Now, under small molecule or ligand-based, we have another strategy that is called Pharmacophore-based design. Pharmacophore-based design, right? Pharmacophore-based design. That means when exactly we should go for Pharmacophore-based design? Say we have molecular docking study. No. Okay, chalo, molecular docking study is not clicking there. Okay, we cannot operate the molecular docking study here. So that means we have with us the biological activity, but unfortunately we don't have the receptor with us. The situation is, in which situation you will opt for Pharmacophore-based design? Pharmacophore-based design. Once you have found that you don't have the receptor with you, you don't have the protein with you in the protein data bank, but you have with you the biological activity of what? The small ligands, the small molecules, the ligands. In that case, you can go for Pharmacophore-based design. Pharmacophore is what? Pharmacophore is what? The functional group. In a raw sense, we can say the functional groups which are responsible for the biological response, the pharmacological activity. Okay? The functional group. That means simply the functional groups? No. If we just try to define pharmacophore, how it appears, it's nothing but the spatial arrangement of what? Of certain features or a set of key features. Okay? Which out of their interaction, which definitely should do some favorable interaction with what? With the active site residues, and thereby emerge or thereby evolve what? Evolve a therapeutic effect, and that is more often that is found to be a beneficial therapeutic effect. So that means not only the functional group which is responsible, the functional group at their respective places, when these two criteria matches, then only we can say those groups are nothing but the part and parcel of the entire pharmacophore. So the spatial arrangement of certain key features, a set of key features which are participating in the process of interaction and thereby emerge, thereby evolve what? Thereby evolve pharmacological response. So that we are going to see in Pharmacophore-based design or Pharmacophore modeling, right? So with that we are done with what? The different strategies or the few strategies, okay, which are adapted in drug design. So with that I am concluding today's discussion. Now in my next video, in part 2, I'll deal with the physicochemical properties, that means the 2D QAs here, which are affecting the biological activity, right? Chalo. You have enjoyed the session, and if you like my video, please press the like button, and please keep your valuable comments, right? Send your valuable comments, and please do subscribe my channel. Okay, it's very much necessary for me to grow. Okay, so thank you for your patience, hearing and all. Thank you.